am at Wake Forest School of Medicine. I work directly under Steve Krzyzewski and have been working the last 2 years under the Executive Committee for the trial targeting aging with Metformin, better known as the TAME trial, and that's specifically what I'm going to talk to you about today. And so that is a clinical trial targeting aging to extend healthy lifespan. As mentioned, I work with Steve Krzyzewski. I also work with Mark Espelond, another biostatistician and co-PI of the trial and executive committee member. But this trial is really the brainchild of Nir Barzilai, who's going to speak next, who he's really invigorated the field in this emerging discipline of geroscience. And George is also involved in the trial as well. He is a key member of our steering committee, and when the trial is initiated, will also be a PI for one of the clinical sites. All right. Okay, and so one thing I guess I should say before we really get started is this is not an ongoing trial. This is a proposed trial. So we are very much still in development, and so what I'm going to share with you today are some of the developmental steps, some of the background, the rationale, and we very much anticipate that many of the details of the trial will probably change between now and the time that it's initiated, but we still think it is an important discussion. And so there are many different strategies when looking at aging. A typical dogma is that we can target a single mechanism, and that one mechanism would, if it breaks down, then there is pathophysiology impairment and eventually overt disease. And if we develop drugs to target that specific mechanisms, we can either prevent, delay, or treat one disease at a time. And though I don't think any of these diseases really happen in a silo at any age, this is especially not true in aging. Aging, as we know, is typified by an increase in the incidence of many chronic conditions together. This is perhaps best evidenced here, but this is some data from the U.S. Vital Statistics showing the cause of death for each of these different disease categories. Each different colored line is a different chronic disease. And what's very evident is that cause of death isn't really monopolized by any one disease. Rather, with advancing age, there's an increase in the death by any number of these, so that aging really seems to cut across these different mechanisms, different diseases. And that's really at the heart of the geroscience hypothesis. Under this hypothesis, we say that we can actually target some of these basic aging processes to delay or prevent chronic diseases collectively rather than one at a time. This hypothesis has probably been most convincingly tested by the National Institutes of Aging Interventions Testing Program. This was already alluded to by George. This is a multi-institutional study that investigates treatments with the potential to extend lifespan and delay disease and dysfunction in mice. The trials have already looked at numerous pharmaceuticals, nutraceuticals, foods, different diets, other supplements, and and they've already shown some efficacy. That in 16 of the trials that have been looked at for lifespan, 5 of these actually have shown an extension in lifespan in at least one sex of mice. These are the nordihydrochloric acid, or NDGA, aspirin, rapamycin, which George has already shown is the most robust, that shows lifespan extension in both male and female mice, acarbose, and 17-alpha estradiol. So in each of these, administration of these different compounds actually extends the survival curve outward, thereby extending median lifespan. The ITP was really envisioned as a large step forward in a translational scientific process that would be used to screen preclinical interventions that could then be translated to humans. But in order for translation to humans to really be relevant, there are many other considerations than just their effect on lifespan. First, these agents, we need to make sure that they're safe. They have an excellent safety profile, that they don't have that many off-target effects. And of course, expense is also important. And so that's really how we arrived at looking at metformin. So metformin is a biguanide class. It was discovered in the 1920s and introduced in the US in 1995. It's currently the most widely prescribed anti-diabetic drug in the world. It is an insulin sensitizer that decreases hepatic gluconeogenesis. And what's great about metformin is that it has wonderful off-target effects. It actually can extend healthy lifespan. And this, while not shown in the interventions testing program, there were some studies looking at lifespan using a metformin dose that's relatively similar to what would be used in humans. And in male C57BL6 mice, when metformin is administered in these adult mice, there's about a 6% extension in lifespan in male mice. However, there's a much more robust health span benefit. In this one study alone, they found that there was preserved body weight, there was no difference in body composition, so that would be lean to fat mass ratio. There was a shift in energy homeostasis and increased use of lipid as an energy source. There was a reduction in cataract development, improved general fitness and physical performance, most strikingly shown by the time achieved on a rotarod treadmill, so improved endurance. Not unsurprisingly, there was a lower HbA1c, fasting insulin, home IR, and lipid profiles. There was a shift in gene expression towards that of younger animals or animals that were on a calorically restricted diet. AMPK was activated, reduced oxidative stress, and there was an inhibition of chronic inflammation. And so that was just one study in mice. And so there might have been a rather modest improvement in lifespan, but there was about a 25% overall improvement in health span. And that's not isolated. This has now been shown in cells, worms, flies, mice, rats, and non-human primates that metformin has many different actions that can target the biology of aging. Outside of the cell, it can interact with receptors for, interact with receptors for cytokines, insulin, IGF-1, adiponectin. Intracellularly, metformin has a number of different actions. It activates AMPK, it has different anti-inflammatory type properties, and it also indirectly inhibits mTOR, which again, George has already mentioned, is a major target for rapamycin and is really well known within this longevity extension paradigm. So all of these effects converge down on the major hallmarks of biologic age, including inflammation, cell survival, stress defense, autophagy, and protein synthesis, which then confer the extension in both health span and longevity with metformin. And so that's how we arrived at metformin, is that we know that it does modulate those critical pathways in the biology of aging, so it can be used to target aging to delay or prevent disease. It has demonstrated effects in humans, both in preventing type 2 diabetes, but also cardiovascular disease in humans. Different observational studies show that it is associated with lower risk of cancer and cognitive decline, and it has an excellent safety profile. It's been used for over 60 years, and also important, it is available as a generic drug, and it's really inexpensive at about a penny a pill, and yet it has all these really profound effects. And so that's why we've centered around metformin as a tool that we think can target aging. Which leads us to our next large question and a lot of effort that we've spent over the last 2 years is trying to figure out actually what a trial on aging would look like. You know, what kind of outcome do we have that convincingly tells us that we've targeted aging? And key among this was actually not just to test this Geroscience hypothesis, but another key goal of the trial was to, work with the FDA to hopefully by the end of the trial have a new indication for aging in drug development. And so we looked at a number of different outcomes. These included many of the things that George has already very convincingly talked about, from different cellular molecular biomarkers, different phenotypes of aging, functional deficits. We explored major mobility disability and have actually come back around and are re-examining that as an outcome. We looked at the possibility of different geriatric syndromes, including frailty or different falls or activities of daily living, emergence of age-related disease, and mortality. Each of these possible outcomes exist on a continuum with gene expression on one end and these biomarkers and phenotypes, and then on the other end, age-related disease and mortality. And along this continuum, there's an increase in the amount of time that would be required to see changes in these different outcomes, as well as the expense of the trial and other parameters. But again, a key goal was to look to the FDA. And the FDA has interest within a fairly narrow bandwidth that is really centered around the hard clinical outcomes, including emergence of age-related disease and also lower mortality rate. And so that's the area that we really focused a lot of our attention in trying to develop. And that's how we arrived also with guidance from FDA through a collaborative and consulting relationship, is that we're looking now closer at a multiple morbidity composite outcome. And so we think that if a drug's effect is on aging, then it should reduce the occurrence of multiple diseases, including those that share very few risk factors other than age. So these need to be from very diverse morbidity families, disease families. We envision this as a collection that would be a composite endpoint of these multiple age-related morbidities, and the primary outcome of interest is the time to occurrence of one of a collection of disease endpoints. The different disease endpoints that would be included in this composite, we have several criteria that were used to determine what these may be. First, the different diseases need to be age-related. They should also be common in the population and pose a significant health burden. Important for adjudication in a clinical trial is that these need to be discrete and clinically apparent outcomes. And again, importantly, they need to cut across these major disease families. And we began looking at this by examining different incidents of de novo multimorbidity. This is data that I'm showing from the Rochester Epidemiology Project, which they use medical record linkage systems to look at the age and sex-specific incidence rates of 20 chronic conditions that had been identified by the U.S. Department of Health and Human Services. So on this slide, I'm only showing 12. On the next slide, I'll show the remaining 8. But what should be really evident is that across all of these conditions, the majority of them do increase in incidence with age in both men and women. This includes things like hyperlipidemia, hypertension, depression, diabetes, arthritis, cancer, cardiac arrhythmia, coronary artery disease, COPD, and osteoporosis. Others include chronic kidney disease, stroke, congestive heart failure, and dementia. There were several that actually did not show these age-related increases in incidence. Important to the crowd that's here today, one of these that did not show an age-related increase was HIV. And so it was no longer considered part of our age-related composite endpoint. Now, because we were also interested in looking at diseases from different families, we were able to go through and sort of bend some of those chronic conditions that increase with age into different families, and came up with a composite outcome that consisted of different cardiovascular type events, including myocardial infarction, stroke, and congestive heart failure. We have cancer that is with non-melanoma skin cancer excluded, MCIs and dementia, and also all-cause death. And our original idea is that we actually had type 2 diabetes included in this composite outcome. However, in consultation with the FDA, this was later removed on their suggestion. However, when the trial is actually initiated, we still do plan to assess type 2 diabetes as part of some sensitivity analyses together with coronary revascularization and also recurrence of pre-enrollment events. Using that composite outcome, we could actually look to some large trials to get a sense of what kind of sample size we would need and what kind of duration we would need to actually see change in these events over time. And so looking at the LIFE trial, which again, George has already talked about, he set me up beautifully. The LIFE trial was a randomized controlled trial of physical activity or exercise in older adults who were aged 65 to 80, the same ages proposed here. And they had slow gaits, or actually they had a mobility limitation at the trial onset. And looking at these data, there's a 5-year projected event rate of almost 11%. In a healthier cohort from the Health ABC study, again, those same two entry criteria, there would be a 5-year event rate of about 8.3%. And so we envision a trial that I would have this sort of multi-morbidity composite, that we would have a very conservative projected event rate of about 7.5%. And again, that's looking only at age and slow gait speed as primary entry criteria. And gait speed was particularly attractive because it's both very easy to assess and it very effectively increases the risk of developing one of these diseases within that 5-year period. So back to our different components, now that we've identified some of the diseases that may be of interest, the next part of building rationale for this trial were then to see, is this composite, is it modifiable? And importantly, would it be modifiable with metformin? And though there are a couple of randomized controlled trials, including the Diabetes Prevention Program and the UK PDS, that give us some insight onto metformin's effect in diseases of aging that are not diabetes. For simplicity today, I'm just going to show you some observational data because that's data that we have at least on each one of these chronic conditions or diseases of aging. And there's been a lot of activity around this recently. This is a meta-analysis that came out, I believe, within the last month or two. And they actually looked at, here, this one is metformin in persons with type 2 diabetes versus non-diabetics. And they're showing an actual beneficial effect of metformin in type 2 diabetes, a reduced there's an effect size so that there's a reduced risk of, this was all-cause mortality. And pulling out one of these studies, just to give a little bit of granularity, this is by Bannister in 2014, is that again, they show here in this green line, this is the cumulative survival for persons with type 2 diabetes who are taking metformin as monotherapy, compared to here in the red and black lines, these are controls, so these are persons without diabetes. And there's actually an improvement in survival in type 2 diabetics on metformin, whether compared to non-diabetics or persons on sulfonylurea monotherapy. And again, they extended this meta-analysis to also include comparisons to type 2 diabetics who are taking metformin versus type 2 diabetics who are taking anything but metformin. And once again, there was a general benefit in mortality in each of these different non-metformin conditions, like altogether, diet, insulin, or other treatment. If we look at different observational studies of metformin and cardiovascular disease outcomes, and if we look at these are here as compared to other either non-use or other agents that were used, that there's a general benefit in cardiovascular disease-related outcomes of around 20%. In persons who actually had some sort of cardiovascular disease at entry, there was also a general benefit on the low end of about 8%, on the upper end of about 24% effect size on reduced risk of mortality. And looking at association between metformin and either cancer incidence, shown over here, or cancer-related mortality, here's another meta-analysis. And once again, there's a general benefit of metformin overall compared to non-metformin use. And the data on cognitive decline and dementia are actually much less consistent. In general, there are many studies that do show a net benefit of metformin on things like cognitive decline as measured by the mini-mental state exam or on dementia diagnoses. However, there were some trials that showing in the opposite direction that metformin was either had no effect or might have actually had a worse risk for some of these outcomes as compared to non-use. However, there was a lot of bias that goes into these, especially when looking strictly at observational trials, which can make some of it difficult to interpret. But I just show this loosely to you to suggest that metformin does have many associations with some of the things that we're interested in the TAME trial, including prevention of type 2 diabetes, and that again has also been shown with the Diabetes Prevention Program and other large-scale randomized controlled trials, prevention of cardiovascular diseases, prevention of cancer is known based on largely observational data. Prevention of dementia or mild cognitive impairment is less conclusive, but there is again an overall reduction in mortality. And so if we pull all of this collectively, looking at the randomized controlled trials, looking at some of these observational data, and combining that with what's known in the preclinical literature, we can anticipate a metformin intervention effect of between 20 to 25%. And so if we overlay that on our event rates that were projected, that's essentially what we'd hypothesize over a 5-year trial. And so that brings us to some of our primary criteria for the trial, and that larger design is that we propose a randomized double-blind randomized placebo-controlled trial. We would use a dose of about 750 milligrams extended-release tablets twice a day. The median follow-up time is proposed to be approximately 45 months. And what's really important is that given those event rates, with this multi-morbidity composite, the trial itself is actually a lot smaller and more feasible than many other prevention trials, with a total sample size projected at about 3,000. And so this was version 1.0 of the TAME trial. Many of these outcomes, criteria, and specifics are being reevaluated right now. And again, the primary entry criteria as originally proposed are ages 65 to 80 and a slow walking speed. 3,000 people would be needed to detect a change in our originally conceived primary outcome that was the time to occurrence of composite major age-related disease from cardiovascular diseases like myocardial infarction, stroke, congestive heart failure, also cancers, MCI, dementia, or death. We had a secondary outcome that was no less important to the initiators of the trial that was really functionally minded, that was major decline in mobility or cognitive function or onset of severe ADLs. And going back to those 20 chronic conditions proposed by the Department of Health and Human Services, we had envisioned a tertiary aim that would look at the rate of accumulation of those. And so just zooming back, though the specifics are going to change, and there may be some changes in the primary versus secondary outcomes or exactly what components go into each one of these. Broadly, we envision TAME to have several different key aims. One would be clinical, and those hard clinical endpoints could then be used in our efforts to create a new indication with guidance from the FDA. We envision another aim that would be really functionally based. And again, a third that's really important for the Geroscience hypothesis, which is to look more broadly at multimorbidity accumulation. And also important is engagement. And so this would be through developing a large biorepository, through courting different ancillaries to look at different phenotypes and biomarkers of aging and being able to tie those back to these hard clinical endpoints. And there would be a lot of outreach also within the trial. And so in summary, TAME is the result of a 24-month process to develop an approach that provides a pathway for FDA approval for drugs targeting aging. We actually think that TAME is feasible and much smaller than many previous prevention trials. And it has the potential to link biomarkers and intermediate outcomes with hard clinical endpoints, paving the way for biomarker use in registration trials of future compounds. Thank you.